All right, good morning, everybody. We're going to do things analog today because the projector is not working. Bulb's out. So I'm going to be writing up here. Hopefully everybody has their digital note-taking workflow down because hopefully you're going to be copying down what I'm drawing on the board. I'll go through the topics as they're presented in the PDF for today, so it should go pretty smoothly. I might have to freehand some meiosis or something like that, but that would be fine. Do we have any questions before we get going? By the way, there is a Socrative quiz like normal, room 240820. Questions? Yes? Are you still recording? I am recording this, but since I'm going to be drawing on the whiteboard, it's not going to show up on the movie. So yes, my voice will be there with the slides and nothing annotated on the slides. Everything is going to be hand annotated on this old piece of technology. At least it's not a chalkboard. Not that I hate chalkboards, but messy. Although I've ruined more pairs of clothes with dry erase markers than with chalk. Any other questions? So I want to clear up one potential point of confusion from last class. So on the second slide, I put back the plot from one of the manuscripts that we've looked at in the past. We were talking last class about sequencing polar bodies to try to determine the sequence of, or the genotype of an oocyte. When I was drawing meiosis, I imagined that there was a diploid organism that went through DNA synthesis, which means there are going to be 4 copies of this one homologous chromosome. So let's say one chromosome is from dad and one is from mom, D and M respectively. We get 2 copies from dad, 2 copies from mom, post-synthesis. Then we enter meiosis. And which is the first division? Which 2 go together? We're going to take a vote. Who say it's one dad and one mom into each of the 2 cells? What about the 2 dads into one, the 2 dad chromosomes into one daughter cell and the 2 moms into the other daughter cell? Okay. Nobody seems to be confident about this, but that's fine. Right, so the 2 versions of the chromosome from the same parent divide, separate into the first 2 cells. Now, if this is spermatogenesis, what happens next?
One stays behind, and then the other one divides.
So how many meiotic products do you get out of spermatogenesis? 4. I saw one person say 4. So you get a second division. You get a chromosome from dad and a chromosome from mom. 4 homologous chromosomes, all versions of the same chromosome. They segregate independently into these 4 different meiotic products. You get 4 functional sperm. How does this differ in oogenesis? I screwed up last time. I gave you bad information. I said it was exactly the same in oogenesis. What's the difference between oogenesis and spermatogenesis? Bless you.
You only have on one side, and the other ones are polar bodies.
So what happens after the first division?
One polar body.
One of these is a polar body. What goes in here? What chromosomes go in the top daughter cell? Guess. The moms. Okay. So that means that the dad versions of the chromosomes go down there. Now what happens? So this is the difference between spermatogenesis and oogenesis. What's the difference? This is the polar body. But what does this cell do? It divides. What are these two things? One of them better be an egg, an oocyte. What's the other one? It's another polar body. So this is the first polar body after the first division, and the second polar body after the second division. So you get one primary oocyte, you get two polar bodies. I believe, if you look back at the video from last class, I showed this polar body dividing. It does not. So the first polar body stays, what's its ploidy? How many copies of each chromosome does it have? As is drawn here. Two. So it's diploid. And the second polar body has a single chromosome of each of the chromosomes in it. So it's haploid, like the oocyte. So what's the big difference between...
I was gonna ask, like, what's the, is it a hormone, or like a hormone change that makes one a polar body and the other
I don't know what makes which one, how the choice is made, which one is the polar body and which one becomes the oocyte. It's a good question. I'll have to, somebody remind me to look that up after class. Excellent question. What's the difference then between spermatogenesis and oogenesis? Well, one, you get all functional gametes out of this. You only get one out of oogenesis. What's the other difference? So the egg is larger. Sperm are motile, small. That makes the people that produce the sperm male. Right? Large immotile gamete is produced by females. How many cells do you wind up getting out of meiosis in spermatogenesis? How many do you get out of oogenesis?
aren't the polar bodies connected?
One polar body, first polar body, second polar body, oocyte. 3. So you get 4 functional sperm gametes. You get 1 functional gamete out of oogenesis, the oocyte. But oogenesis produces 3 cells, spermatogenesis produces 4. Questions about that? That takes us to slide 4 if you're following along. If you arrived late, the projector is not working, so we're doing everything analog, not digital today. So today's core concepts. This week we're doing Mendelian inheritance. And today we do Mendel's first law. Next class we'll tackle Mendel's second law. On Friday we'll do statistics. So you'll probably notice there are 2 reading assignments for this chapter in LearnSmart right now. The second assignment, which is due Thursday night, is just the section in this chapter on statistics. So Mendel's first law today, second law Wednesday, statistics on Friday. We'll have covered this whole chapter. So today, by the end of the day, we get to Punnett squares. Don't black out just because I said Punnett squares. So half of you stopped paying attention when I said Punnett squares because you know Punnett squares, or you think you know Punnett squares. The other half of you blacked out because you don't want to know about Punnett squares. Hopefully I'm going to reach everybody today and convince you that you don't know everything about Punnett squares, or that you actually can understand what the hell Punnett squares are all about and how to set one up and complete it. But to introduce the concept, now I'm on to slide 5. Central dogma. So what's the idea of the central dogma? We go from what's on the left, DNA, that is your genotype, produces RNA, which produces protein. And what do proteins establish for us? Our phenotypes. So one of the entry quiz questions today was, what are some phenotypes you see in this room? Not surprisingly, common. Most of the answers were, I see different eye colors, skin colors, hair colors. Let's see, was there anything more interesting than that? Those are all human phenotypes, visible traits. Tall and short, widow's peaks, hair, eye, blonde. There are a couple of us, yeah, it's true. So, the insight that Mendel had, which is why he is the king of inheritance, transmission genetics, is that your phenotype, while dictated by your genotype, the chromosomes you inherit, the mutations that you have in your DNA produce different proteins which make us all unique. You can also go back the other direction. Your phenotype can tell you something about your genotype. So one of my phenotypes is I'm colorblind. And I have a hard time distinguishing red and green from brown, which I'll show you some interesting simulations of at some point perhaps. So I can use that information that my kindergarten teacher learned when she said, color this thing red, and I grabbed a brown crayon and colored it brown, right? She made a diagnosis. She could have been a doctor. She said, well, she didn't say. She could have said, you have a mutation in your red opsin gene because you don't know the difference between green, brown, and red, right? The next slide, picture of two girls, vertical picture, right? The girl on top has really thick neck. This is symptomatic of Turner syndrome. Individuals that have one X chromosome, not two X chromosomes. So they're female, but they have some phenotypes, some physical symptoms that make them look different. And this is what physicians do all the time. They look at patients, and you can take somebody's phenotype. In some situations, you can look at somebody's phenotype and deduce something about their DNA. So you see this particular suite of symptoms, and you say, okay, this individual probably is missing an X chromosome. So the central dogma works in one direction, but we can use phenotype to guess, to deduce individuals' characteristics about their DNA. So, next slide, the one with the loaf of dog. About pure breeding. That's a purebred dog. I don't know what kind of breed it is. Right? So this is the one piece of terminology that's really important to appreciate and to understand what Mendel's talking about. And it looks like the vast majority of the class, based on the pre-quiz, the pre-class quiz, understands the concept of pure breeding. Okay. So you take an individual, if it's a P as shown here, you can cross individual P's together or you can self a P. So the cross diagram, use an X with an arrow. If this is the F1 generation that's produced from a cross, what does F stand for? Something that scientists made up because we're mean and evil. Filial. So it's the first filial generation, or F1. What's the previous generation? The parental, or P, sometimes P0. I write P0. You can just write P if you want to. The parental generation. Parental, or P0. So you can take two P's maybe one wrinkly and one smooth pea, and you produce an F1 generation. This can be done by crossing with the X and the arrow, or you could take an individual pea, because it's a pea and they're hermaphrodites, and you could self it. A curly arrow or an arrow with a circle next to it, selfing. So pure breeding individuals, if you take two individuals or self an individual, you produce F1 that are entirely the same looking as the parents. So if you go to the next slide, picture of round and wrinkled seeds and yellow and green seeds, two of the traits Mendel looked at. What will happen, what do you predict will happen if you cross the yellow pea and the green pea? What should happen? Not what does happen, what should happen. Go ahead.
There would be more yellow seeds.
Why would there be more yellow seeds?
Yellow would be dominant.
Is yellow dominant? You've got way more information than I need right now. So we've got, no, that's fine though. So dominance. So if you didn't know anything about genetics, what would happen if you crossed a yellow pea and a green pea? What did Mendel think would happen? You'd get half, so half yellow, half green. Each pea is half yellow, half green? Right down the middle? It could, no, I'm serious. This was over 100 years ago, Mendel. Who knows? I mean, they had empirical evidence. They had crossed peas before. They had grown peas. They knew that you didn't get peas that were half green and half yellow. But yes, absolutely. So you might expect in one pea pod you get 50% of the peas are yellow and 50% are green. What else might happen? What other possibilities? The colors mix. So you get, my least favorite color is yellow-green or green-yellow, depending on which crayon you're using. Right? You get a mixture of the two colors, which we know happens in some plants, just not peas. So there were a lot of possible outcomes of Mendel's cross. So that's what should potentially happen. So if you go to the next screen, hang on just a second. I need to launch this Google exercise. So in the next couple of seconds, you should be getting a Google Sheet to your Google Classroom. And what that contains is some of Mendel's data. So I want you to briefly take maybe 5 minutes or so, 3 to 5 minutes. I've put up the data. Okay. So Mendel saw approximately 3 of one trait for every one individual that had a different trait. And based entirely on these values, the 3 to 1 ratio, 3 green to 1 yellow, 3 round to 1 wrinkled, he made a number of inferences. So this is slide 10 in the PDF, page 10. Over 100 years ago, a monk, who his peers thought was probably crazy because he spent all his time counting peas, collected these numbers and made a number of absolutely accurate predictions about how biology works just based on looking at peas. This is biology in every species, not just peas. Every diploid species, anyway. So he came up with this list of 5 inferences. That peas had some sort of hereditary factors, genes, and that every pea could have 2 versions of a gene. So what do we call those versions of genes? Those are alleles, okay? Critical point. What is the maximum number of alleles that any one of us humans can have in a cell? Is it 1? Who thinks 1? Maximum number of versions of a gene we can have in our cells. 1? 2? 4? 16? No. How many copies of every chromosome do we have? 2. So we can have 2 versions max because we have chromosome 1, for example. Doesn't have to be chromosome 1, I just like the number 1. We have 2 chromosomes, one we inherited from our mom and one we inherited from our dads. So we can have a maximum of 2 versions of the same gene. Let's call it the Y gene. So if we've got a pure breeding P, what's the other allele going to be? We have an allele here that's big Y. If it's pure breeding or homozygous, what's the other allele? Big Y. So homozygous from the word same. Right. How do we make a P that has the maximum number of versions of the yellow or Y gene that can have? What genotype would I write down here? We're going to write a heterozygote. Okay, big Y, little y, right? Diploid can only have 2 versions of the yellow gene. Any individual can only have 2 versions. So we've got genes, we've got alleles, or versions of a gene. In this case, we have 2 that we're looking at, a big Y and a little y. Here's something really, really important. Very important. How many versions of the Y gene can there be? How many versions of the Y gene, how many alleles of this gene could exist in nature? 2, 3, depends. Stupid question for me to ask. A huge amount. So it depends on a lot of factors, but the number of alleles that exist in nature of any gene, everybody's allergic to peas. Every number of alleles is essentially limitless. But any one individual only gets a maximum of 2 of those versions because we're diploid. Peas are diploid too. So the number of alleles that exist is limitless. Number of alleles in a diploid, 2. So the reason that we get stuck in this idea of there's 2 alleles is because we often work on diploid organisms and we're used to just thinking about an individual having 2 alleles. And it's so convenient to Western society because we happen to use an alphabet that's got uppercase and lowercase letters that we always use an uppercase to depict one version of that gene and a lowercase letter to depict the other. In some cases, you might see me work on situations where you've got 3 versions, so I would just use numbers. Y1, Y2, there could be a 3rd version, Y3. So there can be multiple versions of a gene, we only work on 2 at a time. Yep.
So the last class we talked about some of that as like a trisomy as triploid.
Right.
For that chromosome, could that be a gene that would have 3 alleles for a certain?
Very good question that lets us apply what we know about meiosis. If you have a triploid individual, could that individual have 3 alleles of a gene that's on the chromosome we have 3 copies of?
No, because that one of them would have to come from the mother and daughter, but one of them would be the same.
Right. So in trisomy, that would be the case where you might have the oocyte. If there's non-disjunction, you've got 2 copies of dad's chromosome, and that fuses with a sperm that is haploid for that chromosome. You get 3 copies of that chromosome, but the 2 that non-disjoined are identical. So those would have to, by definition, because those were produced by DNA replication to have the same allele, which is what you came up with right after you asked the question. So in Mendel terms, those two chromosomes that are in a polar body or in a non-disjoined oocyte have to have the same allele. You could have a different allele in a sperm, like a little y. Great question. Okay, so Mendel's first law has entirely and only to do with math. And it's really easy math, so don't worry. Hitchhiker's Guide to the Galaxy, right? Don't panic. If a diploid individual can have two alleles, so here we go back to post-synthesis, pre-meiosis. Let's say we're working on a P that looks like this. What do we call this? Would this be homozygous or heterozygous? It's got big Y, and little y. So this is meiotic product from a heterozygote. So at the previous step, there would have been a big Y chromosome and a little y chromosome. They undergo synthesis. How do these alleles, the big Y's and the little y's, divide among the meiotic products? We're going to have an output of 4 from this one meiosis. So we've done this before. The eventual products are there's a Y, big Y, little y, and a little y. And I always use these horizontal short lines just to indicate the position on the chromosome of the gene that I'm talking about. So don't worry that they're crosses. It's a chromosome where I'm just marking the position of where I'm saying that that gene exists. Will you ever see from one meiosis, one big Y and 3 little y's? If we start from this, where there's a maximum of 2 alleles, so here's our maximum of 2, a big Y and a little y, replication happens. So what frequency of big Y's do we have in this cell? What percentage of the total chromosomes are big Y? 50%. What percentage of the gametes have big Y's? 50%. There's no other mathematical way to do it. If you take one thing that has an even number of alleles, 2, and you divide those 4 chromatids into 4 separate pools, you're always going to have 50% of gametes with big Y, and 50% are always going to carry the second one. What happens if we do this with a homozygote? If we had a homozygote, and that went through synthesis, meiosis 1, meiosis 2, the 2 divisions, we produce 4 gametes. They're all going to have big Y's, right? There's no other choice. So if you work in a diploid, and you're looking at one individual, this is all the choices you get about what their gametes look like. If you're homozygous for an allele, all of your gametes have that version of that gene on the right. If you're a heterozygote, half your gametes have one version, half of the gametes have the other, 50% of each. Why is this useful?
Allows us to determine the genotype of that.
this knowledge lets us predict what should happen if we mate individuals together. So let's say we did that. Let's say we took a heterozygote individual and a homozygote, and we mated these two together. Let's make a Punnett square. Well, let's not call it a Punnett square. Let's just diagram it. So you've got this individual. What are you going to tell me? We've just talked about this. What will you tell me about, if you describe in words, the gametes of this individual? Do my band apply this?
100% big Y.
They're 100% big Y. So from parent, let's call this parent B, parent A. So parent A was a heterozygote, parent B is a homozygote. Parent B has 100% big Y gametes. So we're guaranteed that every time we cross these two individuals, let's say these are the oocytes, just arbitrarily, we know we're going to get one chromosome that's big Y. Every single oocyte that this individual produces is going to have a big Y. What about parent A? We've got 50% big Y, 50% little y. How many different combinations of those gametes, the haplotypes, the alleles, can be made in this specific case? How many different types of offspring can there be? So we've got two different genotypes. You either get big Y with one of these, or you get a big Y with one of the little y sperm that come from the dad, parent A. How would we draw this as a Punnett square? This is a Punnett square. It's just a diagram to show us all the possible combinations of gametes that come from one parent and the other.
You know, put the genotypes of the parents on the sides. Be like,
So how many rows and columns should I make a Punnett square for this example? So everyone says 2 rows and 2 columns. What do the number of rows and columns in a Punnett square really tell us? What do they represent?
The possible gametes that that parent can produce.
Right, so rows and columns, number of rows and columns, the number of haplotypes, which is fancy for genotype of a haploid cell, right? Haplotype. What's a haploid cell? A gamete. So we're talking about the genotypes of gametes when we talk about haplotypes. So big Y is a haplotype, little y is a haplotype. So the number of rows and columns represent the different types of haplotypes that the parents produce. So what do I want to put on the x-axis, let's say? Which parent do you want to put on this? Parent A or parent B? It doesn't matter.
B.
B, okay. So put parent B on the x-axis. So parent B produced all big Y oocytes. Right. So what do I put here? Big Y, big Y. My question to you is why? Why do we have 2 columns here? Because this individual is diploid. So the number of rows and columns really has to do with ploidy. If you're a diploid, you put 2 rows and 2 columns. What would you do if you were working with a tetraploid organism? You put 4 rows and 4 columns, because what's the maximum number of alleles that you could have if you're tetraploid? If you got 4 versions of a chromosome, you could have 4 different versions of the same gene at the same time. We normally work in diploids, so everybody's used to drawing a 2 by 2 Punnett square. What do we put on this axis? That would be parent A's haplotypes, so gamete haplotypes. What version of that gene is carried in the gametes? What do we have? Big Y, small y. So this is doing exactly the same thing as this. We're just looking at, if you fill those two, as you know, you fill down, you fill across, and you see all the different combinations of genotypes you wind up with in the next generation. So you get a big Y with a big Y, big Y with a big Y, big Y with little y, big Y with little y. Did we really need 2 columns? Why not? You're right. We don't.
This is the same.
It's the same. Big Y on the left column, big Y on the right column. Why even waste your time writing this right column? What frequency of big Y genotypes are there in the next generation? What percentage? We've got big Y, big Y, big Y, big Y, and then the heterozygous. So what frequency of the homozygous do we have? 50%, half. Now what? Did the answer change? No. So why waste your time writing that whole other column of the Punnett square? If you've got an individual that only has 1 haplotype, they're making all big Ys, just write it once. Now you've made a Punnett rectangle.
Dr. Ross?
Yeah.
I think we do the 2 columns because it just makes it more visually pleasing, since we think of the male meiosis where we get 4 sperm cells, where it's more of a, you have to do some math to realize how many of those gametes actually carry that. It didn't turn out to be 50% or something.
So now if you wanted to write a Punnett square that reflected the gametes that are produced by meiosis, what would we do? Here, I'll put it back the way it was. How big would our Punnett square be if we put all of the gametes on the Punnett square? Do you guys? And this is just making more work for ourselves, because now we've got a 16 cell Punnett square, right? I've put every one of these, big Y, little y, on one axis. And I've put 4 big Ys on the x-axis. So let's not make extra work for us. Your Punnett square only needs to be as large as the number of haplotypes. One individual's homozygous, you could only just do 1 column or 1 row, depending on which way you've organized where you put B and where you put A.
Dr. Ross?
Yeah.
I found it like in I guess the questions or the examples they're giving in the book, I found it difficult when they gave us, we were looking at 2 genotypes, and then one of the 2 genotypes was a heterozygous. So then you did have to end up doing that, so.
Like this, we've got 1
Yeah,
heterozygote.
No, they were looking at 2 different traits, so you end up getting like 4 different alleles, and that was very
Right.
more
So-
confusing.
When we start talking about more than 1 gene, that's next class, into Friday, Wednesday into Friday. Then our Punnett square has to get bigger. But when we're talking about a cross that only involves 1 trait, what's that called? Monohybrid cross. So this is a monohybrid cross. We're making hybrid individuals. We're just looking at 1 gene, in this case the Y gene. When we do a dihybrid cross, we're looking at 2 genes, Mendel's second law, then things go a little bit crazier.
Can you just like forget about the dihybrid Punnett square and just make two monohybrids?
So, to briefly answer your question, the dihybrid cross Punnett square is just a combination, like multiplying two monohybrid cross Punnett squares together. We'll get into that. Next class. Okay, so we've talked about how to arrive at the number of columns, number of rows to put in a Punnett square. How many axes? Why don't we draw a three-dimensional Punnett square? That's easy. This is slide 14. We only need two axes because we're sexually reproducing species and there are only two parents. If we ever come up with some sort of biological situation where there are three parents that all contribute to an offspring, we'll have to add a third axis. We'll have to do a Punnett cube. Fortunately, we don't have that as far as I know. Prove me wrong. So the last few slides in the slide deck, starting on slide 15, I said monohybrid Punnett square, what happens if you self a roll minus minus worm? What happens if you cross it to a wild type worm? That would be a plus over plus worm. And what are the next generation's genotypes and phenotypes? I'll leave that for you as practice for next class, if you feel like you need practice doing the monohybrid crosses. This is an example monohybrid cross. Same with the next slide. What happens if you self a GFP asterisk over plus worm? What happens if you cross that individual to a wild type individual? Do that as practice, if you like. The third to last slide, slide 17, get more practice if you want. If you don't do it, it'll show up on the study guide for the next exam, so you might as well do it now. This is about dominance. So in the last 2 minutes, let's talk briefly about dominance. On the pre-class quiz, the most mixed result, your feedback to me about your knowledge of dominance, this was the least consistent response question. So how do you determine which trait, which allele is dominant? If you have big Y's and you have, say, little y's, how do you figure out which one's dominant? So if you mate these two individuals together, a big Y homozygote and a little y homozygote, what's the genotype of their offspring? Definitely. It's got a big Y from that individual, a little y from that individual. So they're heterozygous, F1 offspring. The phenotype that this individual has, that is the trait that they have, tells you which is dominant. Do they look like that parent? Then that parent's allele is dominant. Or does this individual look like that parent? If this heterozygote looks like a little y plant, then little y is dominant. So with that information, you should be able to work on this slide that says cross-training with the plants, with the leaves growing out of the flowers, which is a really weird thing. So practice that as well. I'm asking you to do all this practicing because there's no reading assignment for next class, because we're still working on Mendel's 2 laws. We've talked about the first law today. We'll talk about the second law next time. So when we come together next class, and thank you, by the way, for all of you that already participated in how you cross roller worms and GFP worms together to make roller GFP green fluorescent worms. A lot of you showed me really good evidence that you understand the material, but made some mistakes. Minor ones, but important ones. That combining of two traits, whether or not you're green fluorescent and whether or not you're a roller, is what we talk about next class in independent assortment, Mendel's second law. So we're going to talk more about that process and that specific question that you've already worked on in class next time. So thank you for participating.